very nice to be here. So I left the office a little bit late this morning, and those of you who've been following the weather, it was a slight bit rainy on the way up, so I dropped behind time. I left Madison at 11:15, if anybody's interested. Subsequently, just pulling out from a gas station, I thought I'd accelerate a bit fast, so I got a speeding ticket. I don't know if that's refundable in anywhere, sense or form. I did at least try to talk the officer. Usually I say, I'm from England, I don't understand all this stuff, you know, about speeding tickets, because we don't have them in England, but he didn't fall for that one, unfortunately. Right, so I'm going to start the introduction of the topic for today. Thanks, Mark, for inviting me up. I'm going to try and be easy on you at the beginning. I'm going to keep it political, obviously, all the way through with the current climate. I approve of everything that I'm going to say here. I'd like to introduce the Stem Cell and Regenerative Medicine Center in Wisconsin, University of Wisconsin. We're very proud of the center. You'll see John Orderico on here somewhere, and also Otacharo up here. There's John. And we're part of a community on campus, really trying to foster interactions between people on campus working in this area and faculty and students. And it's a really lively group, and we're proud to be part of the university. If you want to find out more about us, you can go to stemcells.wisc.edu. So my topic today is neural and talking about the brain. I'm going to start with Neuroscience 101, get people up to speed, and then I'll move on to talking about transplantation and how that's impacted my field of neurology. I'll then move to stem cells and replacing neurons in the brain, and then maybe tell you about the idea of not just replacing neurons, but replacing support cells and how that is probably far closer to the clinic. And finally, I'll talk about a potential clinical trial that we're considering. All through the talk, and I won't go into this because you're going to get a lot of this afternoon from Cynthia and Alta, I'll basically highlight some of the ethical challenges. Of course, we're using embryonic research, which is discussed at many pubs around the world every evening. We're discussing human-animal chimeras, which in my field is very important. We're putting cells sometimes into primates or monkeys that are human. We're talking about how many of those cells we can put in before we run into ethical difficulties. And even moving to the clinic, of course, you face ethical trials. So this field, for me, is a little bit of an ethical minefield. And so it's very nice to be having the ethical side afterwards. I wouldn't need to talk too much about that. If you want to have a fun website for you and your family to go to, determinedtoheal.org. I've discovered this recently. It tells you all about what's in the body. We're made up of billions and billions of cells. And the cells, basically, it's amazing that we don't just all fall apart. The cells stick together, and that's what makes multicellular organisms work. And the cells bond together in different lumps. And you're going to hear today about pancreas cells from John, and I'm going to focus on brain cells. But ultimately, every tissue is made of cells in your body. So the idea of replacing cells is what stem cell therapy is talking about and what all the fuss is about. This is very different from giving drugs to patients which treat the cells that are going wrong. We're talking about maybe replacing cells in your tissues and body. And the brain is the area that I'm interested in. This is my son, Sam Svendsen, who you'll meet again. And he used to hear me going to talks when he was 4 or 5 years old, and I used to ask him, he used to say, "What's in the brain, Dad?" And he said he knew, and this is Mr. Brainy, his cartoon character form that he used to tell me he knew what was going on. And then as he got older, he's now 16, he got a little more sophisticated, said, "That was all wrong, Dad. Actually, what's in the brain, I found this on the computer." And I'm like, "Very clever, honey. You know, well done. You found a skull. So obviously that's the outside of the brain." He said, "No, Dad, there's more." And then he did this. I've never worked out how he does this in PowerPoint. But anyway, he said it rotates and does all this. This is what kids do to you. They keep you educated and keep you up to speed. But actually, it's a good description of what's in the brain. And our problem, unlike the heart or the pancreas, is the brain is immensely complicated. In fact, so complicated, something's gone wrong. Interesting. Sam, help. Okay, that's interesting. That's never happened before. Okay. Maybe I'll try it again. Okay. Well, let's just assume that the brain is there. Here we go. It should finish this off. Inside your head is the brain, and the brain is made up of many different areas and regions. So when we talk about stem cell therapy for the brain, you have to talk about very complex tissues, and there's no one region of the brain that's similar to another one. So if you're thinking about replacing cells, you've got billions of different cells, and each area of the brain has a different structure and anatomy. So it's quite a challenge to think about repairing cells or putting cells into the brain. Now, within the brain itself, most of you probably know about the neurons, which are the cells that connect one part of the brain to the other. But there are two other characters I want to tell you about. First is it's a horrible name, oligodendrocyte, and the second one is called an astrocyte. And these are two support cells in the brain. And it's rather like a hospital. These characters in the brain, these cells provide support for the all-important neuron. So the neuron needs these cells to survive. What do? The oligodendrocyte actually ensheaths and wraps around the wiring of the brain, all these long fibers that you have. It's rather like the insulation in the house for the wires. And I'll tell you about what happens if this dissolves as a disease called multiple sclerosis. So if you lose that insulation, you get MS. So you can automatically think about replacing these cells that die in MS with stem cell-derived tissues. But the other cell I'll talk about is the astrocyte. Now, these cells sit absolutely alongside the neurons and control most of what the neurons do. And the astrocyte is a little understood cell. Nobody talks about it, but there are actually 3 times more astrocytes in your brain than neurons. And if those of you know Einstein, when he died, his brain was kept in a jar. They've looked at it recently, and they asked, well, he must have had more neurons than everybody else because he's Einstein. Actually, he had more astrocytes, these support cells. He had 3 times more astrocytes than the normal person. So astrocytes are actually quite important, although we don't quite know how they got Einstein to be so clever. I've certainly injected some and it hasn't had any effect. Okay, these are the nursing cells. Now, these are the cells that we're dealing with, and now I'll step up a little bit and go forward into what goes wrong in these cells. We have a number of different diseases, Alzheimer's disease, and they're usually characterized by different people that get them, just to make them familiar to you. This is a loss of memory. Cells die widely throughout the brain, particularly in the hippocampus and cortex, areas associated with thought and memory. Stroke, Margaret Thatcher suffered a stroke some time ago. This is actually one of the leading killers in America right now, I think second only to heart attack. And stroke is where you lose blood supply to a small part of the brain, and the neurons in that area die because you've lost blood supply, usually through an embolism or clot. Multiple sclerosis, I just mentioned, is a lack of myelin around the cells. Huntington's disease is a genetic disorder. Woody Guthrie had this, a folk singer, and that's genetically inherited as a neurological disease that's passed on through families in a very determined way. And of course, blindness, retinal degeneration is something that's very common. Macular degeneration also is another disease of the brain. We consider the eye an extension of the brain. And the two I'm going to talk about more, I will come back to blindness briefly, is Parkinson's disease, and this is Pope Jean-Paul, Muhammad Ali, and of course Michael J. Fox, who started a foundation up for working in this area. And amyotrophic lateral sclerosis, ALS, Lou Gehrig, the baseball player, had this. So this is how complicated brain science is and how many different diseases we have to deal with. And if you think about stem cell therapy, you can't just say stem cells for the brain. You have to actually go specifically and look what stem cells can do in each of these different neurological disorders. And I, of course, don't have time today to go through each one in detail, but I will just start with Parkinson's because that seems to be the one that always gets into the newspaper. When you talk about stem cells, it's like, oh, well, that's a cure for Parkinson's disease. I'd like to say, first of all, that I don't think it's a cure right now for Parkinson's disease. I think that's very difficult in this disorder, and I'll explain why. But I think stem cells may provide an interesting treatment for Parkinson's disease, just like we have treatments today. Cure is a very strong word. It means you're going to reverse the symptoms, you're going to stop the disease, and the patients are going to completely recover. I'm not sure stem cells are a miracle cure for Parkinson's or other neurodegenerative diseases at this stage. However, they do have very interesting implications for treatments. In Parkinson's, you get the stoop gait, you lose cells. This black area is called the substantia nigra, where the dopamine neurons live in the brain. And the cells die, and this patient died, and you can see there's no black pigment left in this part of the brain. This is what the brain looks like. Here's the cells that die in Parkinson's. They have a long projection up here into this area called the striatum. And the chemical that these cells produce is sent up this long cable and is released here, and the chemical is called dopamine. And for some reason, these cells here die in Parkinson's. We don't understand why they die, but they die in Parkinson's disease. And this results in a loss of dopamine up here. And it's this chemical that actually is required, rather like the oil in the engine, is required for you to have a very slick movement. Now, we've asked this chap in England, and you'll see him again later on in the clinical trial that we did, to do this as fast as he can with his hand. Turn it over as fast as he possibly can. And you can see how slowly he's going. You can see the classic resting tremor of Parkinson's disease that these people have. And he's unable to move, and he's unable to move because the oil's missing in his engine. In his brain, he doesn't have enough dopamine because the dopamine neurons have died. So the question is, can we transplant dopamine neurons back? So give him new dopamine neurons, and maybe he'll get better, and we'll cure Parkinson's. This is a very simple idea, cell therapy, cell replacement. And it brings up the interesting question of, is brain transplantation possible? Now, who's seen Steve Martin in the movie The Man with Two Brains? So he had a screw-top technique. Okay. Unfortunately, that doesn't work very well, but actually as long ago as 1564 in England, which is where I'm from, a gentleman otherwise well, this is English surgery, very old surgery in the 1500s. He went to the king, this guy, begging him to command the physician, king surgeon in ordinary and myself, to open his head, remove his diseased brain, and replace it with another. We did many things to him, but it was impossible for us to restore his brain. This is the first ever attempt at whole brain transplants. I wouldn't recommend this. Obviously, there are complex issues with replacing a whole brain, unlike the kidney. You can replace a whole kidney, perhaps a pancreas. A whole brain is difficult. You wouldn't quite be the same when you came out of the surgery. Obviously, this didn't work. He looks very old, but actually, if you look carefully, he has a laptop computer right here. He's emailing home for his dinner. Always throw that. Apologies, anybody who's seen me give this talk. So the message is adult brain transplants don't work. So a number of years ago, groups in Sweden started exploring a different strategy. And in rats and mice, they said, what if we take the brain when it's developing into dopamine neurons? So they went back to fetal tissue from the rat, in this case, a 14-day-old fetus from a rat, unborn fetus. They removed the fetal tissue in the brain, and they took out the embryonic part of the brain that was going to make dopamine neurons in that animal. They took them out, they put them into little pieces, they digested them in a tube, they then put them in a syringe, and they injected them into the brain of rats that they'd given experimental Parkinson's disease to. And the result was quite miraculous. Nobody expected this. Here's a stain of a rat in a cross-section. Here are the dopamine neurons on one side, and they've gone on this side of the brain and the spinal cord. Here you can see the result is no dopamine. This is a marker for dopamine. And here's a transplant of this fetal dopamine neurons. Can you see how they've gone in, they survived, and they're now releasing dopamine in the rat brain? And this was the first evidence that you can do a brain transplant. It is possible. But of course, they're not transplanting the whole brain. They're taking fetal tissue, a small part that's responsible for making dopamine neurons, and putting it in the brain. This led to the News of the World is a very respected paper in England. It's equivalent to the National Enquirer. I'll just put it there. However, they reported on this first trial done in humans by a group of Uli Lindvall in Sweden. And this is the first ever human brain transplant. And even back then, they decided the tremendous potential of the treatment of humans outweighed any doubts over using material from aborted babies. And this material came from abortions at this point in Sweden. So this is the ethical debate already opening up in 1987 when they started doing this type of therapy for humans, and they had to use a fetal tissue. What happened? This is the cross-section through a patient. What you're looking at is a PET scan showing live dopamine levels. This glowing blob here is dopamine, and on this side of the brain, he didn't have a transplant, and this patient is getting a lot of dopamine into the brain from the fetal neurons. So they've gone in the brain, they survived. Those of you can see up here, the neurosurgeon drills a hole in the brain in order to inject the cells. So this is quite standard surgery, and over 350 patients have had these transplants, and they're actually doing quite well. Some of these patients are still alive 14 to 15 years after receiving a transplant. The brain is immune privileged, so you don't have to give immune suppressants continually. You can actually remove immune suppression after 6 months, and the brain transplants will survive, unlike the pancreas and other tissues in the body. And there are some long-term effects. Now, unfortunately, this hasn't turned out from all these fetal tissues to be a cure for Parkinson's. There are a number of challenges, some of which are very detailed, and many of which neurologists have been arguing over for the last 10 years. There are sort of people for this transplant therapy, and there are people against it. And one of the major issues is we do these transplants in patients, they're called ectopic. You're putting them back in a part of the brain where the fibers died, whereas the actual neurons that transmitted this dopamine were way down in the brainstem. So these are ectopic transplants. What happens if you put the neurons down here? Well, this is an old age problem in the brain. They don't regenerate axons all the way back up to reinnervate the striatum. So if you put the cells down here, they're pretty useless because they don't make the dopamine in the right place. They'll survive, but they won't complete the circuit. So this is a real roadblock for Parkinson's disease. The other problem in Parkinson's is there's more systems than dopamine neurons that degenerate. So the patients that have transplants show other loss of neurons in the other parts of the brain. And perhaps the most problematic is once these transplants are in, they'll keep functioning even when you don't want them to. So if anybody saw Michael J. Fox getting, some people said he was making out movements on television some time ago. That's called a dyskinesia movement due to the medication that you take for Parkinson's. And that dyskinesia, when you take your medication, can be induced by the transplant. The transplant makes dyskinesias, and because it's in the wrong location and it's not completing the circuitry, the dyskinesias can often be uncontrollable. So this has been a real roadblock for clinical trials in Parkinson's. So even though we have fetal tissue, people have stopped going ahead with these types of drafts. But the idea holds great potential for Parkinson's. I've just given you a reality check on where we are now. The idea still holds a lot of potential. And I think where we're headed is, if you watch here, the magic will occur. What we'd like to do is put the dopamine neurons here and encourage them to grow back the circuit to release the dopamine up here. And there are experiments underway to try and get that going. So we're moving in that direction. There's a slight pause right now in Parkinson's, but if we could get there, that would be great. The real problem is ethical again and getting enough tissue. So fetal tissue is very unreliable. There's no control over the cells you put in. Patients end up with uncontrolled movements if they're in the wrong place of cells. And of course, the ethical concerns. Some countries you can't even use fetal tissue in Ireland, for instance. So we need other sources of tissue. So now I get to the stem cells. I wanted to fill you a background of Parkinson's to put you in a framework of where stem cells are coming in for Parkinson's disease. And of course, the first idea is to replace, again, the dopamine neurons. So what can stem cells do for this? Well, if we just introduce you to stem cells briefly, you've all heard of stem cells from all different parts of the body. Stem cells are all over the place. You get them from actually the adult brain has stem cells, the skin, the bone marrow, even the fat. We're very lucky that the fat you can isolate and take stem cells from. The fetal brain that I just talked about has stem cells, and of course, embryonic stem cells you hear more about during the day. Embryonic stem cells are the most primitive. They come from the inner cell mass, this small cluster of cells, at 8 days of development. After the sperm fertilizes the egg, makes this small ball of cells at 4 or 5 days, and there you isolate the embryonic stem cells. And Jamie Thompson in the University of Wisconsin was one of the first to do this. Wherever you get these cells from, you then have to, in my field, make neural progenitor cells, and that's been quite difficult from adult tissue. However, it's very easy from fetal brain or from embryonic stem cells, and I'll leave that discussion right there. So the course really is you have to really use embryonic or fetal tissue in order to get neural tissue. And at the end of the talk, I'll mention an exciting new technology where you can derive pluripotent cells from these adult tissues, but that's only been available very recently. So you expand these cells, and then we want to push them down to neurons, oligodendrocytes, and astrocytes that I've talked about. I'll mention in a minute that along the way, the nice thing about using stem cells rather than fetal tissue is you can engineer the cells, you can modify them, you can change them before you put them into the patient. You could maybe make them produce regulated dopamine release, so you can switch on and off the dopamine production in the Parkinson's brain by giving him a second drug. That's the excitement. This is the transplantation of these cells that we do, and of course, eventually we want to go into humans. And I went through these diseases earlier on, the range of diseases that are being discussed. Back to Parkinson's, Suchan Zhang in the lab next to mine in the University of Wisconsin successfully took these embryonic stem cells. Now, here's an interesting picture. So you take embryonic stem cells, which just look like amorphous cells in the dish, and you push them a certain way with a cocktail of growth factors from the fridge. This is all in the culture dish. You can make them into neural tissue that looks like this. Can you see here little tubes, little round tubes? Each one of those is like a neural tube, which is how your brain starts to form. So you can push them into brain structures by giving them the right signals. Cells from the embryonic stem cells are pluripotent. They go in any direction. You'll hear about pancreas in a minute. Here we're pushing them neural. So each one of these is a little neural tube. And then if you keep pushing them, you get these beautiful dopamine neurons that you can see in this picture. So we can produce dopamine neurons. We can actually transplant these into the rat brain, into the monkey brain. And what we're trying to do is control now the release of dopamine. So we engineer these dopamine neurons so they're responsive to a second drug. Now we can put the cells in the patient or the rat or the monkey, and they won't make dopamine until the patient takes another drug. It's called an inducible promoter system that we're engineering in. In that way, it'll be a lot safer to try these transplants in patients. And these other problems, of course, remain, but now we have a source of tissue to make dopamine neurons. We're slowly starting to address these problems with neuron replacement in Parkinson's disease. But it's quite a long trail yet from there to the clinic. I think we're going to take a pause. We have to work out the reconnection, and we have to work out the dopamine release. And of course, embryonic stem cells, that you hear from John, are inherently dangerous. With great power comes great responsibility. If we put them in the brain, and there's one embryonic stem cell left, it may start dividing and trying to make tooth, bone, hair. It's called a teratoma. And so you don't want that in a patient. You definitely don't want it in the spinal cord where there's limited space. It could cause a lot of damage. So we have to be careful about using these cells for clinical trials. So just to broaden out a little bit in the last part of the talk, I'm going to discuss the potential of using stem cells for the brain. Now clearly we take the stem cell, we can differentiate them into these three different neural types. I've talked already about an example of making neurons, sort of the brain hero if you like. But these need integration, and they have to make correct long-term connections. There's a reason why the brain doesn't regenerate in humans. It's because it may be worse for you to have a bad connection than to have no connection at all. And that's something to consider when you're thinking about neuron replacement in the brain. Okay? The brain is very adaptable. Most of us in here only use 10% of their brain. That's the common myth. So you have 90% that you maybe could re-energize and redirect in. Okay? Hopefully you're using 70% right now. The oligodendrocyte that I mentioned before, the myelating cell, is being thought about. We can get that from embryonic stem cells for trying to get a therapy for MS, and there are people like Ian Duncan on campus at UW who are pioneering this. The trick there is the lesions in MS, the damage, occur all over the brain, and it's where how you can target them. And there's a group at Geron in California who are considering using this derivative for spinal cord injury. These are patients that have injuries to the spinal cord, and remyelinating some of those fibers may get enough signal down the spinal cord to get some functional recovery back. And those clinical trials are supposedly close. Maybe Cynthia knows more about it than I do. Every year since 2004, they've been saying the clinical trial will start next year. And they're still saying the clinical trial will start next year. The question is which year? So hopefully that'll start soon, but they're being very cautious, and the FDA is obviously worried about teratoma formation from these cells. Now finally, this poor guy here, the astrocyte that nobody ever talks about. So this is my favorite cell. Astrocytes might modulate many disease processes. They're easy to transplant, and I'll mention now they could also be used to deliver drugs to the brain. And getting drugs into the brain is often a challenge. The blood-brain barrier, many of you may have heard of, limits the amount of drug that gets into the brain. 97% of compounds can't cross the blood-brain barrier and are useless to pharmaceutical companies. So this is an important issue. And one drug this is my hype-hope slide. I think new neurons in the brain is kind of hype at the moment. We're a long way from getting that to work. However, the hope part, and I hope this will work very soon, is the astrocyte replacement and drug delivery. And this is where I'm going to in the next section. And the drug I want to talk about is called GDNF. It's called Glial derived neurotrophic factor. This is discovered in 1992 by a company called Synergen. Synergen was eaten at by Amgen. Amgen got all the patents, so they now own the patents to GDNF. We know that this drug protects dying dopamine neurons in the brain. It also it enhances, it's like fertilizer. Those of you looking at your lawn now, you probably went out at the weekend to Menards and were in line to get fertilizer for the grass. This is like a fertilizer for brain fibers in the brain. And if the brain senses GDNF, that's usually there during development, those brain cells will become more functional. And in fact, it may prevent them from dying. In monkey models of Parkinson's, GDNF has trophic effects and stops those neurons from dying. But as I mentioned, it's very difficult to get GDNF into the brain itself. So back to our model again. The idea now is neuroprotection. Can we protect? Dopamine neurons are lost in Parkinson's, but when you when you first get a diagnosis, you have a lot of time window in order to protect the ongoing death. So the question now is, can we, when your patient first presents to the clinic, can we get in there with a neuroprotective strategy of his or her remaining dopamine neurons? And what I'm going to show you briefly is a clinical trial that we performed in England before I left for the US, where we infused with Amgen GDNF, this growth factor, directly into the brain to avoid the blood-brain barrier. And so the idea is that GDNF will now get to these dopamine fibers that are dying in Parkinson's. And the beauty of this is that the fibers will take GDNF back into the dopamine neurons down here. So they transport the protein all the way back to the Niagara, where the dopamine neurons are. So this is a really cool therapy, because you get back labeling into here. You don't need to inject it all the way down here. It gets sucked into the neuron and sucked back. So Steve Gill, a colleague of mine, a neurosurgeon friend, who I met giving a talk rather like this, and in a British tradition, we went to the pub afterwards and had a discussion. We had stem cells, and he had neurosurgical skills. So he wanted to put stem cells in the brain. This is like 9, 10 years ago. And I said, well, GDNF is much more doable now. And so we started this project. What he did is implanted catheters into 5 patients, and you can see them here. And attached to the catheter under the skin is a Medtronic's pump that goes under the stomach here, under the stomach wall. And what we do is turn this pump on with a magnet from the outside filled with GDNF. And through these small catheters, the GDNF is infused directly into the brain of the Parkinson's patient. And this first clinical trial was done in England. This is the same patient that I showed you right at the beginning of the talk. And this is the patient after his GDNF treatment. And this is how he should be doing this task of continually turning his hand, and he goes through a series of other tasks. So the GDNF treatment off medication had a significant effect in this patient and in the 4 other patients that were in this clinical trial. Now, after this, I don't have time to go into what was the nightmare after we published this paper in Nature Medicine, but this is called interactions with industry. And Amgen got so excited about this data and these patients, they ended up doing their own trial using a very different method of delivery of the GDNF, and it ended up not quite working. They had some effect, but not enough. So they shut down GDNF. There are a number of patients suing Amgen right now. Amgen are not very healthy at the moment, and in fact, I just heard they're probably selling a lot of their rights to GDNF, so maybe it will come back into the clinic again. These are scans of our patients, and you can see here, this is after the GDNF and before. So not only did we see the patients respond, when we looked at the brain, it had more dopamine. This is the area of the brain that makes dopamine. On average, we had significant increases in dopamine levels in the patients that received GDNF. Now, because of Amgen's troubles and complications of pumps, this is where the stem cells come in. We engineered stem cells to make GDNF, and the idea now is, rather than having a metallic pump that you have to pay Amgen $2,000 a day for the drug, which is what they were charging at that point, I said, well, why not give the patients a drug for free? Right? They didn't seem to understand that concept. What does free mean? America doesn't, it's a free country, but everything costs money. I've never understood that. I said, the drugs can actually make, GDNF can be made by cells. And once we make the cells and put them in the brain, you have one surgery, the cells go in through a catheter, and once they're in the brain, they'll pump that drug out for as long as you live, if the gene is switched on and maintains on. So you can get free drug for the rest of your life. So a great concept, unless you're a drug company. We have to figure out how to make money from this. That doesn't worry me. I'm an academic, so I can just say all this stuff. We don't need dopamine neurons for this therapy, because remember, we're adding a drug that protects your own dopamine neurons. So now we have the problem of the reconnections gone. We actually started this work about 10 years ago, and we started with fetal tissue because that was what was accessible then, that was left over from some of those clinical transplants with the Swedish neurosurgeons. We developed a way to grow these cells in the dish, and then in collaboration with colleagues in Switzerland, we put viruses on, and you'll hear about viruses again because they're used to induce pluripotent cells. We put viruses on these neural stem cells from the fetal brain, and then the cells would be engineered. The virus delivered the GDNF gene directly to the cells, and now we had stem cells that pumped out GDNF. They're GDNF-releasing stem cells. This was done in collaboration with a group in Switzerland, Patrick Abischer, and a postdoc in my lab, Bernard Schneider. So we have the cells, they're releasing GDNF. We also, just recently, in this summer, we'll have the final bank made. In discussions with the FDA, said, well, can we make banks of these cells, stem cells, that will be able to release GDNF? We call them progenitor cells because they're not really like an embryonic stem cell. They're further down the path to neural. So they're neural progenitor cells. They make GDNF, and Derek High at the Clinical Biomanufacturing Facility at the Weizmann Center, where I work, now has this up to clinical grade level. So we have hundreds of tubes of these cells that are ready to go into patients by the summer. This is what we do, and I've shown you this before. Take the GDNF secreting cells, we put them into a syringe and inject them into our models of Parkinson's disease. And if you're looking at here is a marker for human cells. Remember, this is now a chimeric rat brain, and this is where Alta and Cynthia come in. We've made a chimera, we've made a human rat species, if you like, and the brain now is infiltrated with human cells. And one of the questions we're always asked is, well, how does a rat start thinking like a human if you put enough cells in? And these are the issues of chimerism that maybe will be discussed during the day. But these human cells integrate. Every black dot is a human cell. And actually, this was amazing integration. And the person who did this study, Shana Burstock, I was so excited by this data, I ended up marrying her, and that's Shana on a winning day. And then just to bring my son back into the story, that's Sam, who did the initial picture for you. So Shana went on, and these are the cells in 3 different animals. This is showing now GDNF release. So GDNF is being made by the cells. Shana modified the cells to pump out GDNF. And we can see that they've actually distributed very nicely in the brains of these rats. And now we're getting long-term GDNF production for free within the rat brain. I don't have time to go into all the other data in the rat, but we saw in those rats functional changes that suggested the cells releasing GDNF were blocking the Parkinson's progress. Together with Marina Emborg, we have a new paper coming out this week showing that we can do the same thing in primates, in monkeys, which is a critical preclinical stage before going to patients with Parkinson's disease. Here's a transplant in a monkey brain cells, and this black dot here is showing you the cells make GDNF, the molecule that's protective. And if you look down here, this is a dopamine marker, tyrosine hydroxylase. Wherever the cells are that you can see in the same monkey slice, you get more dopamine. So the stem cells are making dopamine in the monkey and making GDNF. And where the GDNF is, those dopamine neurons are being protected. So now we have primate evidence that this is working. And the monkeys that had the transplant did better in their Parkinson's rating scales. So lots of pieces of the puzzle are coming together. Right? So just to conclude, for Parkinson's disease, and I'll have time to finish on ALS, for the Parkinson's disease, we've shown that cells can be modified to release GDNF, stem cells or progenitors in the monkey brain. They integrate, and they actually form astrocytes. I didn't show you that data. They don't form neurons. They make astrocytes. We think the astrocytes may also support the tissues in the brain just because they're astrocytes and they're the hospital, if you like, and that the GDNF has functional effects. Why are we going into the clinic today? The last step with this process for patients, and for the neurologists in the audience, so you might appreciate this, is we have no control at the moment over the GDNF produced by these cells. They're pumping out GDNF, but we want to have a way to switch it on and off. And I mentioned earlier on, there's inducible promoter systems for dopamine that we're thinking about. We're trying to adapt those for GDNF. So now the patient gets the transplant, nothing happens until they take a second drug which activates the GDNF release. And actually this makes everybody happy because you can charge $100 a day for that second pill to activate the drug release, all right? So Amgen are quite pleased about this, that we have this problem. But anyway, they always win in the end, believe me. You can't do anything. So the last 2 parts, just to quickly go through, this is going to be very quick. The other ideas we have for these cells, it's not just Parkinson's, right? These cells now in the bank can be used for other things, and in collaboration with David Gamm and Ray Lund in California, we've been looking at using these cells in retinal disease. Now, for those of you who are very squirmish, please close your eyes at this point, and then I'll tell you where you can open them. First question is what can they do? And I'm going to show you this is support again. I think close them now. Okay. All right. It's all over now. And for those of you who missed it, there it is again. Aha! I caught you. This was made by David Gamm. I told him, you cannot show this. It's very, very bad, this big needle going. But what David does, and he's a retinal surgeon, and he always tells me, Clive, you've got to work in the eye. He's a surgeon. He said, because if it goes wrong in one eye, the patient always has the other eye. And I'm saying, well, I don't think that's a very good strategy, David. Rather it worked first time. Don't talk with surgeons around beer. It's just not good. So here's a needle. We put the cells in, and this time we're putting them behind the retina. And what we're hoping to do in macular degeneration, and this is a model of, the RCS rat is a model of macular degeneration, which is very common in this country. We hope the cells will go into the eye and regenerate the eye. And in fact, every black dot, this is the retina, Every black dot is a stem cell that's moved into the eye, migrated, and filled up the retina. And actually, when we look at, in detail, at parts of the retina and the survival, this is a part that had no cells, and this is a part that had cells. And when you look in high power, the part that had cells has pretty much got normal retina. The part that didn't have any cells, or the cells didn't get to, has a degenerated retina. And this was, again, published recently. And when we do the functional test, the rats can still see. So the stem cells have gone in, and through some sort of trophic effect or the modulation of your own circuitry, prevented degeneration. And this is a theme which is coming out in stem cell biology, whether it's adult stem cell transplants into patients with heart disease. They're not making new tissues or heart. They're actually modulating the heart disease process. So somehow these cells are neuroprotective or heart protective or even maybe protect pancreas cells from degeneration. This is a common theme in stem cells at the moment. We don't understand a lot of these mechanisms, but seems to be how they work. So I'll finish on Lou Gehrig's disease, which for me is one of the saddest diseases that you can be diagnosed with. It's called motor neuron disease, or ALS. Lou Gehrig is the person who exemplifies this disorder. And what happens is now we're down in the spinal cord, okay? And cells die, just like in Parkinson's disease, in the spinal cord. And this ends up, these cells project to the muscle, you end up losing motor control and losing muscle control. There's no treatment for ALS, unlike Parkinson's disease. The prognosis is death within 4 years. There's one drug called Riluzole that prolongs life by about 3 months. That's it. The strategy here is, just as I've been telling you before, this time we're in the spinal cord. The strategy is when there are still motor neurons which are connected to the muscle, let's get the GDNF secreting cells that I've told you all about, put them in the spinal cord, and see if they'll migrate into the area of damage, which we know they will in other models, and restore the function of these motor neurons. So we've now made a chimeric spinal cord in these sick patients. And the spinal cord has healthy astrocytes, and the astrocytes are making GDNF. And GDNF works just as well, but unbelievably, on motor neurons which drive the muscle, as it does in Parkinson's disease. Sandy Klein, who's a graduate student in my lab, showed that this can work. She took the same process in the spinal cord, put the cells in, and I'll cut to the data. Here are the cells, this time a very dense graft of human cells that make cells that look like astrocytes. Here's a surviving motor neuron in the spinal cord, and every red dot is a human cell that's gone around and protected this rat motor neuron, and subsequently the rat motor neurons survive better. And the cells, just to be sure that they make GDNF, here's another transplant, and here's the cells making GDNF, exactly where the cell transplant went. The other cool thing with this technique is you can target one area of the brain for your drug delivery. It's not going to get everywhere and cause side effects. This is a really important concept, I think, again, for drug delivery and drug therapy. Now here we're not so worried about switching on and off GDNF because you have 4 years to live. You'd rather have a side effect from too much GDNF than death. So those are your choices in ALS. And we get neuroprotection. Here you can see in a transplant where the graph was, we get more motor neurons surviving. So these cells can help the motor neurons survive. So that's an example of 3 examples of different diseases that we're looking at using stem cells for. Neuron replacement is tough. Astrocytes and growth factor release is much easier, we think, to imagine. And that's led us to be as bold as to suggest we can do a clinical trial. And the disease we've chosen is ALS. We've chosen to use cells releasing a growth factor, GDNF, and transplant them into a few regions of the spinal cord where the patient's limbs are fed from. So we'll go into an area of the spinal cord that controls the legs, and we'll look to see if there's an effect on motor neuron degeneration and muscle strength in the legs where we put these cells. Now, you could go to China, or indeed Italy, for programs, and in fact, this China program has now been shut down. This is another stem cell side effect, that if you have $40,000, you can go anywhere in the world and get a stem cell transplant. They never tell you what the transplant is, and the saddest thing is a lot of these patients come back and they think they have an effect, it's called a placebo usually, and then after 3 or 4 weeks, the effect fades off, so they save up another $40,000 and go back for the same stem cell treatment. And unfortunately, a lot of these groups are charlatans. You have to be very careful. We're trying to solve this by doing clinical trials in this country. And this is the clinical trial that I'm involved with, and we're in the process of dealing with the FDA to get approval for putting these cells into patients. And my last slide really just sort of brings up, segues into John's discussion with diabetes. And I think this is a general context for going into patients with stem cells or any treatment. Really, for neurological diseases, we have to get the balance right. So you have to look at the seriousness of the disease and the risk to the patient before starting a clinical trial with anything, and stem cells included. So if the risk of the seriousness of the disease is very high, you can take pretty high risks. And this is where we don't need regulated delivery of the drug for ALS. If it's a medium seriousness of disease, then maybe you need a medium risk. And if it's a low risk, low seriousness, so not a very serious disease, the risk to the patient has to be very low. You wouldn't go in a disease that could be treated easily with a radical stem cell treatment that may cause cancer and kill the patient. So we have to be sensible when we think about disease and stem cell treatment. There's not a panacea. It's very complicated, but it's possible to pull these treatments together and move forward if you pick the right ones, and I think John will discuss diabetes in a moment. Now, of course, this is a favorite slide. There's a lot of debate around using stem cells against people destroying life. There's lots of excess embryos, and they could save lives. But do we need to use embryos? And this is two slides I put in at the end. Again, I forgot I put these in, actually. And John is going to go through this again, so I'll very rapidly tell you about these two papers. We didn't think it was possible to make embryonic stem cells in any other way than go from the oocyte, fertilization, in a cell mass, and then destroy the embryo, or essentially kill it. Recently, two groups, one in Japan, one in Wisconsin, Jamie Thompson's lab, have shown they can do this with human cells. The group in Japan did it with mouse cells about 2 years ago. Now we can do it with human cells. What you do is you take a skin sample, make a fibroblast, which is normally just a normal fibroblast, can make more skin cells, or fibroblast cells, and you hit it with 4 genes. And you reprogram that cell. And this comes back to Dolly the sheep, for those of you who remember. Dolly the sheep was made from an adult cell that was then, through nuclear transfer, made into a whole sheep. Well, this way is cheating. You just take the skin cell and you reprogram it to go back in time by putting these 4 genes in. And remarkably, you get something out of this that looks like an embryonic stem cell line when you express these 4 genes in these skin cells or fibroblasts. So this is now shortcutting everything, and now we can go from here to pluripotent cells without destroying an embryo. Our lab is also interested, and in fact working at the moment with Jamie, on generating patient-specific cell lines. And this is another whole area of medicine which is going to allow us to do this disease in a dish. If we have Parkinson's disease, you take skin cells. Now I can make iPS cells and then dopamine neurons, and we can understand maybe the mechanisms of disease. And I think John will talk about this also for diabetes. And of course, the nice beauty of this as well is that we may be able to do autologous transplants, which means we could take a skin cell from any of you in the audience, take it through this process, and now have an autologous cell, the cell from your body, go back in, but play a different role. All right? We've changed it now from a skin cell, say, you want to cure baldness, to being a hair cell. So now you can regrow your hair from a cell that came from your skin, or we can reprogram it to be a brain cell. Now, all sounds great. There are lots of complications, and John may be going into these more later on, but the potential is there for using these cells for this type of work. But there are complications, and I'm sure we'll be hearing about some of the issues, particularly putting genes into cells may affect them in ways we don't understand yet, so they may not be completely comparable to embryonic stem cells. So we have some work to do there. So to finish, I've finished on ethical points, because hopefully you picked up the ethical nuances as I've been going through the science. We've got to have discussion at all different points. Embryos and fetal tissues raise these issues. I've been working with fetal tissue for over 20 years and been dealing with issues of fetal tissue and embryos that have to be addressed, which is why this meeting's so good. I think we have to look at human suffering and moral concerns, balance those against needs for the patient. iPS cells that I just discussed may change everything. That's a big may. It was a very exciting time right now, and again, we'll be discussing this today. And the take-home message is the first role for stem cells in the clinic for the brain may not be to make new neurons that you all think about, but replace support cells or astrocytes, act as mini-pumps, and protect host neurons from the effects of ALS and Parkinson's disease. One last point, this is an advertisement for, we're hosting the World Stem Cell Summit here in Madison in September 22nd to 23rd at the Alliant Energy Center. It's going to be a big group of people coming from all over the world to discuss stem cells and ethics and policy and advocacy and outreach and the science, of course. This is my little home. When I came here from England, they told me the weather was like it is or was yesterday the whole year round. So I had to draw the little sun. Being a stupid Brit, I believed everybody. So I don't want to go to California. Wisconsin's got the same weather. Why would you bother? My lab's up here. And lots of people worked, lots of people worked to bring this together, and I've mentioned most of them along the way, and I'll leave it there. Thanks very much.